Hello everyone. My name is Haruka Yuasa. I'm a student in Miyata Group from Osaka Metropolitan University in Japan. My theme is structural analysis of MreB filaments causing helicity switching swimming in syn3B. I'm working on spiroplasma motility. The left movie shows spiroplasma. They swim in viscous liquid by helicity switching between left-handed and right-handed. Previously, our group reconstituted spiroplasma swimming in syn3B by expressing two types of MreBs. For example, this is MreB1 and 5 strains, and this is MreB4 and 5 strains. In the current swimming model, two types of MreB make swimming motility. So our focus is to reveal the mechanism of helicity switching from structural perspective. And now we focus on this MreB4 and 5 strain. At first, I talk about the significance of using syn3B for structural study. There are two merits. First one is limited protein complexes. Spiroplasma has filament structure in the cell, and it is composed of fibril and 5 types of MreBs. However, by using syn3B, we can control the combination of MreB, just MreB4 and 5. So we can observe only target structure, MreB4 and 5 filament. Second merit is small. Syn3B is small, so it is to reveal the structure, we use cryo-electron microscopy. So syn3B is small, so it is easy to observe an inner structure as they are. So these are the merits for using syn3B. Next is our strategy. Our focus is to reveal helicity switching mechanism. So we'll do two things. First step is visualization of filaments. For example, how MreB4 and 5 align in swimming syn3B. And next step is to visualize high-resolution structure of filaments. For example, how MreB changes the conformation in a cell, and how MreB4 and 5 interact each other. Today, I focus on this first step, alignment of MreB4 and 5. Please see this light picture. This is MreB5 filaments in vitro. MreB5 makes anti-parallel double filament, and it makes sheet-like structure. And in the spiroplasma, there are filament structure like this in the cell. So from this character, there are many possible patterns of MreB alignments. For example, MreB4 and 5 are adjacent to each other like this. Or this is a pattern 2 in protofilament units like this. And this is pattern 3. MreB5 makes sheet, and MreB4 protofilament is adjacent to the sheet. So to reveal the alignment, we need to distinguish between MreB4 and 5. To distinguish between MreB4 and 5, we use mCherry as a tag. This is MreB4 and 5 mCherry structure. MreB5 has mCherry like this. And this MreB4-5 mCherry strain also can swim like this, like spiroplasma. At first, we check filaments in the cell by cryo-electron tomography. This left movie shows whole view of the cell. And this is magnified image. You can see the sheet-like structure here. You can see sheet-like structure like this schematic. This line means cell membrane, and this is sheet-like structure here. And in this right image, there are filamentous structure around cell membrane. So we successfully observed MreB filaments by using syn3B. Next, we tried visualization of MreB alignments. So we analyzed a detailed filament structure near the membrane. We pick up 66 particles of filament, and they were classified into 5. The results suggested 2-layered filamentous structure along cell membrane. This line 1 and line 2. The monomer pitch in line 2 is approximately 5 nanometer. This is close to spiroplasma's MreB monomer. And the filamentous structure in line 1 cannot distinguish between MreB and M-cherry so far. If this filamentous structure in line 1 is M-cherry, we can say filament in line 2 is MreB5. So more sample is needed to determine it. In conclusion, we successfully observed MreB filaments in syn3B. Our goal is to elucidate the mechanism of helicity switching. However, the number of samples is not enough to reveal the alignments. So now we are planning to increase the number of samples and distinguish MreB4 and 5. Next, we are also planning to solve the high-resolution structure. Now we can observe helical and sheet-like structure like this from syn3B. So I'm working on isolation of this filamentous structure. That is all for my presentation. Thank you for listening.
This is dazzling. Questions for Haruka? So what will you do? What is your next plan with this?
Next plan is take more picture to visualize more fine structure MreB and M-cherry to distinguish MreB and MreB4.
Is there something, and I was looking at one of your earlier slides. So are you putting your M-cherry on the ends of the MreB, or is it in the middle of it?
Yeah, middle of it, MreB.
How did you choose the spot?
Sorry, I can't answer it now. Sorry, I can't answer it.
That's okay. I don't know is a perfectly valid answer. Before we go on to our next talks, I want to thank my Japanese friends in the Miyata group, because remember, folks, that while I'm here at 5 in the morning on a Friday, these folks are giving up their weekend. This is fairly late in the evening on a Saturday night in Japan. So really thank you. And I promise next year I won't have you wrecking your weekends with having to talk on a Saturday night. I wasn't thinking when I set the timing up. Still, this is dazzling work, and I can't wait to see where this takes you. What is this telling you about spiroplasma biology? Last question.
Spiroplasma biology. Sorry. Please say again.
So you are studying spiroplasma motility.
Yes.
What do you now know about spiroplasma motility from these experiments that you didn't know before?
Yeah, it should be the difficult question for him. So then, now we are working on the the possibility of the MreB protein. So then, and we wanted to specify that identify what is the cause of the or origin of the motility. So then the MreB protein is, as you know, so it's not related. It's not involved in any of the motility. It's just the layer structure for the peptide glycan synthesis. So then our interest is what is the origin of the cell motility. So then we are that catching just it's originally MreB protein is not related to the motility, but we can see that just the two the different types of the MreB protein that gives the motility. So then we believe that then we can see that just the kind of the really early stage of the development of the cell motility of the mycoplasmas by the studying about the MreB protein in the simple system of syn3B. That's our understanding.
Okay. Thank you, Makoto. Again, thank you to your group. This is just spectacular work, and it makes me believe more and more in the enormous power that modern imaging has for us.